That is all for me. Thank you very much.
Okay, Sylvia, great. Thanks very much. Again, another fascinating presentation. And again, looking at the clock, I see that we don't have time right now for questions, so we'll ask folks to hold their questions for the panel discussion that'll come up. But again, thanks very much for doing a really nice job. So thank you. Okay, so next we have up is Dr. Alan Boobis. Let me get my notes up here. Alan is Emeritus Professor of Toxicology at Imperial College in London. His research interests include mechanistic toxicology, mode of action, and risk assessment. During his distinguished career, he's published approximately 250 original research papers and sits on a number of national and international advisory committees, including the UK Committee on Toxicity, which he currently chairs. The title of Alan's presentation today is UK Safety Assessment of CBD as a Novel Food. Alan, over to you.
Thank you, Leo. And I'd like to thank Norb and the Chris MSU for the opportunity to talk to you this afternoon about CBD. I'm not a regulator, obviously. I do give advice to regulators, and it's from that perspective that I want to talk about CBD as a novel food in the UK. I have to get yeah. So we've heard a very useful explanation of the various categories which CBD could come into in the US. And whilst there's overlap in Europe and the UK, there are some important differences. And we have this categorization of a novel food, which is a food that has not been widely consumed by people in the UK or Europe before May 1997. So our cutoff date is slightly later than the one in the US. It means that the food does not have a history of consumption in the population. It could be a new food. So some years ago, phytosterols and phytostanols were introduced. They were novel foods. It could be a traditional food that's eaten somewhere in the world, but not in our population. And in recent years, we've seen the introduction of chia seeds and other products. Or it could be a food produced from a new process. So recently, bread treated with UV light to increase vitamin D levels or vitamin D levels was considered novel food. And before a novel food can be legally marketed in the UK, it has to have a pre-market safety assessment and authorization. But the complication is there is no single list of novel foods or a list of foods that are not novel. So that if a company wants to market a food in the UK, there's no easy way of knowing whether it's novel or not, because there's no single source that says, well, this is already on the market or it's never been on the market. So as a consequence, over the years, a number of potentially novel foods, including CBD extracts, have entered the UK food market without prior authorization. And you've heard that these CBD containing extracts include beverages, various oils, chewables, and chocolate. But recently, the UK confirmed that CBD extracts are novel. This was decided in January 2019, and they've now been entered into the EU Novel Food Catalogue, which is a partial list of novel foods in Europe. Now, hemp and related products, such as cold-pressed oils from hemp, are not considered novel because there's evidence to show a history of consumption in the UK prior to May 1997. So one important thing is that if a product's on the market, it can't simply be removed. There has to be some indication of an adverse health effect to have this unauthorized product removed from the UK market. But it's certainly clear that non-medicinal products containing CBD are becoming increasingly popular in the UK as they are elsewhere in the world. And the Food Standards Agency, which is responsible for the regulation of foods in the UK, have been asked about risk assessment of CBD in such products on an increasing number of occasions in the recent past. So the UK process is that for novel foods, their safety assessment and recommendations for authorization are the responsibility of a committee called the Advisory Committee on Novel Foods and Processes, or ACNFP, which is one of the Food Standards Agency's scientific advisory committees. Now, it's important to note here that the ACNFP is responsible for the assessment of a specific food commodity supported by a sponsor. So it's looking at the product that's going to be marketed, not its ingredients individually, although that would obviously comprise part of the assessment. If the CBD contains more than 1 milligram of THC, it would mean that this product cannot be authorized as a novel food and comes with the responsibility of the Home Office under the Misuse of Drugs Act 1971 legislation. So an important point to consider here is that in the UK, CBD is not currently regarded as under the purview of the Misuse of Drugs Act. In other words, although it's a cannabinoid, it's not believed to be psychoactive. So it's distinct from something like THC. Now, there's another committee, the Committee on Toxicity of Chemicals in Food, Consumer Products and Environment, COT, which I chair at the moment, which is jointly managed by the Department of Health and Social Care and the Food Standards Agency. And amongst other things, it's responsible for providing generic advice on chemicals of food, such as CBD, to the Food Standards Agency. So this first step was the FSA came to the COT and asked us to review the available toxicological information on CBD to determine whether there was any problem, and if so, what was the magnitude of the problem. So obviously we were primarily interested in CBD, and as other organizations that reviewed this product, the information was primarily on the medicinal grade CBD greater than 90% purity, which is approved now as a therapeutic drug from GW Pharmaceuticals, known as Epidiolex, and is approved in both the US and in Europe. And I would like to say that the COT is really grateful to GW Pharmaceuticals for actually coming to attend our meeting and discuss with us and share their data. Almost all the information that we reviewed is in the public domain through the submissions to EMA and FDA, but we had additional insight from the pharmaceutical company themselves. So I'm going to review a little bit of the toxicology. You've heard most of this already. Just want to give our COT interpretation and highlight a couple of additional points. So first of all, as you've already heard, CBD is poorly and variably absorbed from the GI tract, and its fasting bioavailability of the pure compound is only around 6.5%. And that increases up to 4-fold in the presence of food. We heard yesterday that novel formulations of CBD are being developed, such as nano-encapsulations, to improve their bioavailability. Now, this is really important when interpreting the toxicological database, because most of the available studies were performed with, let's call it normal CBD. It wasn't manipulated to improve its bioavailability in fasting subjects. So the NOELs or the low-Ls that we see in those as individuals have to be considered in that context, that the actual bioavailability is lower than it could be in a free-living individual, and particularly if you take a new formulation. Some important kinetic considerations, just highlighting the ones that we were concerned about in addition to the information that was provided today and yesterday. It has a relatively slow half-life, about 2 days. It's metabolized primarily in the liver, and the oxidation is by CYP2C19 and CYP3A4, and these are P450 enzymes responsible for a large number of therapeutic drugs. It's also glucuronidated by several UGTs. You've heard the metabolism includes 7-hydroxy-CBD, which does pharmacological activity, but has unknown toxicological effects, and 7-carboxy-CBD, which is not active pharmacologically. In the repeat dose clinical studies, you've seen this several times. These are the side effects that were reported with an incidence of at least 10% compared to the placebo-controlled subjects: elevation of liver enzymes, effects on appetite, otherwise on the GI tract, somnolence and fatigue, insomnia, and other effects on sleep patterns, infections, rash, and drug-drug interactions. And I want to go through the key endpoints here and tell you where we got to at the COT. So in hepatotoxicity, which has been discussed at some length already, in the clinical trials in patients with Dravet syndrome, receiving 5 milligrams per day for 3 weeks, one patient developed ALT at greater than 5 times the upper level of normal, and at higher doses, more patients developed elevated ALT. In a phase 1 study in healthy volunteers, 5 out of 12 subjects receiving 5 mg per kg per day for 3 weeks showed elevations of ALT above normal. In this study, no subjects receiving 1 mg per kg per day showed any elevation of transaminases. But I would point out this is a relatively small number of subjects. In studies in experimental animals, there is some evidence that liver is a target with effects evident at doses of 15 mg per kg or above. But the effects observed, such as increased liver weight, hypertrophy, elevated alkaline phosphatase, some transaminase changes and CYP induction, may have been adaptive and may not be toxicologically significant. And we just don't know. And unfortunately, we did not have access to the raw data on the preclinical safety assessment studies of Epidiolex. We just had the summaries. The CNS effects have been emphasized. Somnolence was the commonest side effect in the clinical trials. It was seen at the lowest dose tested, 5 mg per kg per day. And other CNS depressants, such as alcohol, might exacerbate the CNS effects of CBD, but we have no data on this. I won't dwell on the immune system effects. You've heard several presentations on this. Clearly, CBD at certain doses can affect the immune system, and there was some hint that there might be some increase in infection in patients at doses of 10 mg per kg and above in the clinical trials. But these data are not very secure. Now, on genotoxicity and carcinogenicity, we referred this to our sister committee, the Committee on Mutagenicity, which comprises experts in the evaluation of genotoxicity data. They looked at both CBD, pharmaceutical grade, and food grade. On the CBD of pharmaceutical grade, there was sufficient information on in vitro and in vivo testing to conclude that it was not genotoxic. But the problem with the food grade CBD, where the data were conflicting, and in the absence of adequate in vitro and in vivo data, it was not possible to reach a conclusion on the genotoxic potential of this substance. CBD has shown no evidence of carcinogenicity in a study in rats, and a study in mice is ongoing. And reproductive and developmental effects, again, we heard about that this afternoon. No effects on fertility have been observed in either male or female rats treated with the compound. Embryo-fetal effects were observed in rabbits, but not in rats. And effects included delayed development and effects on the testes. There's no data on whether CBD is present in breast milk. It is a lipophilic substance, and it's possible that there is exposure via this route, but we have no information. Drug-drug interactions in vitro studies have shown that CBD inhibits several forms of P450, including some that are involved in the metabolism of a range of human medicines, CYP1A2, CYP3A4, CYP2C19, and the Ki value is in the micromolar range. And these effects have been confirmed in vivo, that their inhibitory potential of CBD has been observed with interactions observed at doses of 1 milligram per kilogram or less. No lower dose has been tested. So to summarize the tox evaluation by the COT, pure CBD, pharmaceutical grade, has very low bioavailability, but this can be increased by food and by change in formulation. Food grade CBD is a variable composition. And when considering it as a novel food, one has to consider the other components in the product, as well as the CBD itself. And we know that CBD extracts can contain other cannabinoids, amongst other chemicals. So the relevance of the conclusions based on clinical studies of CBD to CBD containing foods is uncertain. It's not possible to reach a conclusion on the genotoxic potential of food grade CBD. CBD has been reported to cause adverse effects on the liver at doses of 5 milligrams per kilogram and possibly lower. The toxicological relevance of these effects is currently not clear, particularly at these low doses. Effects in the CNS have been observed. It can inhibit the metabolism of a number of drugs at doses as low as 1 mg per kg. Effects on the developing fetus and offspring cannot be excluded. And so the FSA, based on our evaluation, concluded that doses of greater than 1 milligram per kilogram per day, equivalent to 70 milligrams in a typical adult, may be associated with adverse effects, but it's not possible in our view to identify a safe dose. So a dose of 1 milligram or lower is not necessarily a safe dose, it's just that a dose above that is potentially a harmful dose. And the URL here shows the location of the review by COT. So what are the next steps? Well, since there is evidence of concern from CBD itself and CBD containing products, the FSA has declared that businesses need to submit validated novel food authorization applications for all their products containing CBD by the 31st of March next year to enable their continuing marketing in the UK. These applications will be reviewed by the ACNFP, and the FSA position can be found at the website shown here. The requirements that sponsors need to meet are the same as those developed by the EFSA, the European Food Standards Agency, Food Safety Authority, sorry, in the contents of this regulation, which have been ported over directly into the UK regulation. And the information required includes characterization of the novel food, including how it's produced, its composition, etc., information on its metabolism and other toxicokinetics in animals and humans, and toxicological information on genotoxicity and repeat dose toxicity up to 90 days, including information on neurotoxicity, immunotoxicity, effects on the reproductive organs, and endocrine-mediated effects. The need for chronic toxicity or carcinogenicity studies will depend upon the results of those subchronic studies. Is there a signal that requires further exploration and longer-term studies? And the same with reproductive and developmental toxicity. Is there information in those subchronic studies, such as endocrine effects or pathological effects in the reproductive organs, suggesting further information in specialized studies would be necessary? And obviously, human data, if available. So I thank you, and I welcome any questions now or in the panel discussion.
Okay, Alan, very good. Thank you very much. Again, another fascinating presentation. I'm going to stop your slide sharing here.
Okay.
And if we can have Ricardo and Sylvia join us, I think if it's okay with everybody, we'll just go ahead and dive into the panel discussion. Nora, but it looked to me like you had a question